Unit 6, Lesson 1, Evidence for Evolution. This is our first lesson in our very last unit. So have your packets ready. Listen, make sure that you're writing down any extra stuff that might be helpful for you to remember later on so that we are learning to be great note takers. Before we start about the evidence, we have to sort of introduce evolution. And we're going to do that by starting off with the father of evolution, Charles Darwin. This man is a just naturalist from England. He went on travels all over the world. He went on the ship called the HMS Beagle. And during his travels, he collected evidence, and this evidence was capturing animals, doing drawings, taking plant specimens, all these different things to then look and compare. And that led him to propose his theory on evolution. And he was observing these characteristics of these animals and plants. And the one place that he found the most differences that really, really just caught his eye were the Galapagos Islands. When he got back to England, he took all of his information, all of his drawings, all of this, and he compiled it into a writing that he published called The Origin of Species. And his current theory of evolution is the theory that is, again, has not been disproven. And he is known as the father of evolution. Here's a map of his travels. Again, he started in England. He went down, around, and back around. And the little yellow spot that's over in the Pacific, that is the Galapagos Islands. And on those islands, he found so many different, again, birds and tortoises that are just similarly enough, but have differences from island to island. And he wanted to know, why were they different from island to island? One of the things that he noticed, and you have that picture of Darwin's finches, is that he noticed that there was a connection between beak shape and size and what they actually ate, and also what food was available on that island specifically. He also noticed that there were tortoises among all the different islands, and depending on the landscape, that actually dictated sort of what shell shape a tortoise might have. He was not the only one, though, to be thinking about how organisms change over time. One was actually during his same time, Alfred Russel Wallace. He was another naturalist. Instead of working with rare and odd organisms that were found around the world, he worked specifically with insects. And he came pretty much to the same conclusions as Darwin. The only difference why he's not the father of evolution is that he didn't publish as quickly as Darwin. Darwin got his work out first. Now, there was a man that also proposed a type of evolution. His name was Lamarck. He came before Wallace and Darwin by almost a century. And his theory said that he thought that organisms evolved via acquired characteristics. And we've talked about acquired characteristics. Acquired characteristics are those traits that you get after you're born, or you develop after you're born. You acquire them through life. You're not born with them. You're not inheriting them from your parents. And he used the giraffe as his main organism of evidence. And he's like, well, the giraffe is able to stretch its neck throughout its lifetime because the trees were tall. But we know that's not how that works. But he didn't get to spend a whole bunch of the time observing these giraffes. So he's like, well, how do they have these long necks? It's really like they're horses. But they grew their necks longer because they needed to get food. And they did that over their lifetime. And the big takeaway from this is that we now know Lamarck is wrong. And Darwin and Wallace helped, again, finalize that by replacing that acquired characteristics with inherited characteristics instead. Now, what is evolution? We're going to define it as the process by which modern organisms have descended from ancient organisms. And there are a couple summary points that Darwin had in his Origin of Species. The first thing is that we know organisms differ and that variations between those organisms are inherited from parents. Organisms produce more offspring than survive. Organisms will produce many offspring, but not all of them will grow up to be adults. That could be due to competition, disease, predation, a lot of things that could, again, contribute to that. We know organisms compete for resources. They compete for food, water, space, mates. We know organisms with advantages survive to pass those advantages to their children. Organisms that have advantages over others are going to survive longer, which means they're going to, again, reproduce more and have more offspring. And those offspring are going to inherit probably that very advantageous trait that made them sort of be able to survive longer than others. And species alive today are descended with modification from common ancestors. So we can look at what's alive today, and we can look at the past fossils and other organisms that we know existed and can see some similarities between them, but they're not exactly the same, which means something has changed. Now, again, evolution is change in a population of organisms over time. It is not individuals. It is population. So mutation is actually the change in an individual. So an individual will mutate. If the mutation sticks and gets passed on into the population, it can cause a population to evolve. But again, populations evolve, not individuals. And there are 4 pieces of evidence that help support this change over time: fossils, comparative anatomy and structures, embryology, and biochemistry. Fossil record, we're going to define this as the preserved remains or traces of an organism that is no longer living. These are going to be, they could be imprintations into rock. They could be an actual bone or something else that's been solidified and not decomposed. There's a lot of different things that we call fossils. But the main thing is they're remains or traces, and they are not alive. And the biggest part of that support is that we're able to date the fossils. In other words, we're able to determine their age. And there's 2 types of ways we do this. The first part is relative dating, where we compare other fossils that are at the same area. So we know that there are layers of rocks, and those layers of rocks form over time. We know that the lower layers are older than the younger layers because of sedimentation, things like that. And if you have a layer of fossils that we know what the date of them, the age of them are, and they're right beside other fossils, we can make that assumption that they're of a similar age. Now, how do we figure out exactly how old something is? We do something called absolute dating, which we chemically test the fossil, and we're testing for radioactive isotopes that allow us to, again, figure out how old that process has been going. Now, the problem with this and why we don't do absolute dating on every single fossil is it's going to destroy the fossil. So they're very picky and choosy on which ones they actually do test. What can fossils tell us? Well, fossils can tell us that organisms have appeared, disappeared, and changed over time. So we know that we have organisms that we can't find fossils of. We know that there are organisms that we have fossils of that don't exist today. And we know that there are organisms that look very similarly to some past fossils, but aren't exactly the same. We can also see an extinction of species. In other words, like I said before, things disappear. We see organisms that used to live that we cannot find in current day alive. We also can look at climate and environmental conditions, specifically ancient ones, by looking at the rocks, looking how different layers, on whether there was a presence of a lot of water or not, whether there were plants, whether there were a lack of plants, what kind of, again, geography we were looking at. We also can indicate a development of life from simple to complex. The oldest fossils that we have are very simplistic. And as we proceed to current day, the more and more complex organisms get. Lastly, there's an indication that life began in water. A lot of our oldest fossils, the lowest levels of the rock layers that we've discovered so far, they have aquatic. They have a lot of like water feed, indications of water. There's the organisms that are there are more aquatic in nature. Now let's talk about comparative anatomy. we're going to define this as the study of anatomical structures to find similarities and differences. In other words, we're comparing organisms' structures. And there are three terms that we're going to focus on. The first one, homologous structures. We know and we remember from a variety of different units that homo means same. So we're just saying structures that are the same. And so this is defined as parts with similar basic structure but different function. In other words, the structure is very similar or the same, but the function is different. And this is very apparent in our image that we have right below. We have a human arm, a cat leg, a whale fin, and a bat wing. All of these have traces of the same types of bones in the same order. They might be stretched out a little bit differently, but we can definitely identify. For example, the radius and ulna are always beside each other, no matter what. And that you have these sort of small bones, the phalanges, that make up the fingers, at least in humans. So there's that similarity in structure, but the function is different. Humans, we use that we use our arms to move around, to grab, to lift. We don't use them to walk like cats do. We don't use them to swim like whales do. And we don't use them to fly like bats do. So the function is different, but the structure is the same. Now we're doing analogous structures. Now remember the AN and A from our very first unit, that means no or without. And so this is the case where we have same structures but different, again, same function but different structures. Again, same function but different structures. They're made differently. So structures that have the same function but different structures, they're made differently. A good example of this is there are so many organisms out there that can fly, but the structures that allow them to do that are completely different. Bats have bones and tissues that connect those bones to form those wings. But the wings of insects are structurally very different. But both of those structures on both of these types of organisms allow them to do the same thing, which is to fly. Lastly, we have vestigial structures. Vestigial structures, they're parts that have little to no function but probably used to be helpful. In other words, they're in the organism's body. They're not really doing anything. They're not really helpful. They don't really harm, but they're not really helpful. And they don't have a current function. But we could assume that at some point in time they probably did. In humans, appendix is one of those. Goosebumps are one of those. Wisdom teeth. There's a lot of things in humans that we have the ability or have this structure, but it's not really helpful to us right now. And sometimes it can even be harmful in the case of appendicitis, when we have to have that actually taken out. But we can live without our appendix, no problem. Embryology. This is the stages of development before the birth of an organism. So it's how an embryo develops, and it's the study of that embryo as it's developing. And this pattern of embryological development, we're going to write down similar patterns of development indicate a common ancestry. So I want you to look at this image over here. You have fish, turtles, chicken, pig, and human. They all start out as a zygote, that single fertilized egg. And as they develop, you'll notice that the first type of develop, they're very similar in their structure and how they're developing, even though those organisms are very different. And as they proceed, they continue to sort of, again, branch out in their differences. But one of the things that we know is that all of these organisms, us included, are in development, have gills. And then in humans, they disappear. They also disappear in pigs, chicken, and turtles. But fish keep them. So that's some sort of similarity, whereas other organisms that are like, for example, not chordates or another different phyla, they don't have those characteristics. So we use embryological development to sort of help group organisms by similarities to say that they have a more common ancestry. We also know that if you look at all of mammals in general, like pigs and humans would be examples of those, that they have a longer string of similarities before they start branching off. And so again, we look at embryos, the longer they stay similar, the closer they're related they may be. Lastly, we have biochemistry. This is proteins, RNA, and DNA molecules, the universality of the genetic code. And the similarities of genetic molecules. The more closely related organisms are, the more similar is the biochemical makeup. In other words, if something's more closely related, they probably are going to have similar like proteins and DNA and things like that. That's why we do DNA testing. And this is going to indicate a common ancestor if, again, you have some similarities overall in your DNA. In other words, if this organism has more similarities with this organism's DNA than another organism. We also know that universal rules of DNA and genetic codes support evolution. So we have evidence of evolution. We've finished our notes, but you are not quite done. You have your practice that you have to get done. So the first part of the practice is going to be identifying the 3 different scientists and their descriptions that match. The second part is going to be utilizing that evidence of evolution to identify the images. And then you're also going to be looking at fossil records. All right, have a great day.